The process of decay. This topic summary explores the process of decay in mammals and its implications for forensic science, particularly in determining the time of death. Stages of decomposition. Fresh stage days 1 to 2. Anaerobic bacteria from the gut spread and start breaking down cells. Blowflies arrive and lay eggs in natural openings and wounds. Bloated stage days 2 to 6. Maggots hatch and feed on tissues, releasing fluids and gases, causing bloating. Body temperature rises due to maggot activity. More blowflies attracted, along with beetles, for example carrion beetles that lay eggs. Parasitic wasps arrive to lay eggs in fly and beetle larvae. Active decay stage days 5 to 11. Maggot masses liquefy tissues, causing strong odors. Fluids leak into the surrounding environment. Beetles and their larvae continue to feed on tissues and maggots. Fly populations decrease as tissues dry out. Advanced decay stage days 10 to 5. Most soft tissues consumed, leaving skin, cartilage and bones. Body dries out significantly. Cheese FLIs and coffin FLIs replace blowflies. Beetle species with strong mouth parts, for example hide beetles, feed on remaining tissues. Dry remains stage days 25 plus. Mainly bones and hair remain. Mites and moth larvae feed on hair. Decomposition slows significantly. Body eventually skeletonizes. Factors affecting decay. Temperature. Higher temperatures accelerate decomposition by increasing the rate of chemical reactions and insect activity. Lower temperatures slow down decomposition. Level of exposure. Exposed body. The internal temperature of an exposed body initially drops below the ambient air temperature due to the cessation of metabolic processes. As decomposition progresses and insect activity increases, the body temperature rises, often exceeding the ambient temperature. This rise is caused by the metabolic heat generated by maggot masses. Eventually, as tissues are consumed and maggot activity decreases, the body temperature returns to ambient levels. Buried body. The internal temperature of a buried body also drops initially, but tends to fluctuate less than an exposed body. The soil acts as insulation, buffering the body from large temperature changes in the environment. The temperature rise due to insect activity is less pronounced in buried bodies. Forensic entomology. This field uses knowledge of insect life cycles to estimate post-mortem intervals (PMI). By identifying the types and developmental stages of insects on a body, forensic entomologists can provide valuable clues about the time of death, research and limitations. Studies using animal models have contributed to our understanding of decomposition, but there are limitations to extrapolating findings directly to humans. Overall, the topic emphasizes the critical role of understanding decomposition processes and insect activity in forensic investigations, particularly for determining the time of death and reconstructing events surrounding a death.